Et sans plus attendre, on va passer à la deuxième partie de la session. On va accueillir deux courtes présentations. Donc Gulia Lioi pour commencer, qui nous vient de Rennes. Gulia est postdoc. Elle a fait sa thèse en connectivité EEG à Southampton, un autre point commun avec Stéphane Demner. Et elle nous présente un protocole combiné EEG-IRM. Donc par rapport à la matinée, on avait l'EEG et IRM relativement séparés. On a ici deux exemples de présentation de protocole neurofeedback clinique en combinaison IRM et EEG, et ici dans la réhabilitation des patients avec AVC. Julia.
Je vais présenter les travaux de l'équipe MPEN et hybride à Rennes sur le neurofeedback bimodal EEG IRMF, en particulier pour la réhabilitation AVC. Je vais faire cette présentation en anglais, mais n'hésitez pas par la suite à vous poser des questions en français aussi. This is the overview of the talk. So I will start by a short introduction about the potential of neurofeedback for stroke rehabilitation, a short point of measuring brain function with EEG and fMRI, because this has been covered already in the previous presentation. I will then describe the bimodal neurofeedback platform that is in place in Rennes, in the CHU of Rennes, and show some results from a pilot study on stroke patients, and conclude with some perspectives. Stroke is a serious health problem, which is the cause of is the leading cause of disability worldwide. It's estimated that 33 million persons per year survive stroke, and 80% of these people are affected by motor disability of one side of the body, typically an upper limb or a leg. And if many therapy are available, such as traditional physiotherapy or robotic-assisted physiotherapy, they have limited recovery outcomes. So most of the time, patients affected by stroke have limited the recovery or very poor recovery after 1 year from the stroke. Literature on plasticity after stroke really put the accent on the importance of relearning some patterns and reactivating a lesioned area in order to have an efficient motor rehabilitation. So in this context, neurofeedback has great potential, and for relearning, for training self-regulation of perilesional areas. And this is an example of how the motor re-education or rehabilitation neurofeedback loop work. So in the first block here, we have motor imagery. So in application intended for motor recovery, the patient is usually asked to perform motor imagery of the affected limb. Motor imagery involves the similar brain circuits or brain areas than the actual execution of the movement. So recording those activities, in this case using integrated EEG and fMRI, and then feeding back this activity to the patient is supposed to help the patient to learn self-regulation of those affected areas. So there are different approaches for neurofeedback rehabilitation for stroke, and Stefan has described some of them, but the characteristic of these approaches is that they rely on one imaging technology. So we have EEG neurofeedback approaches or BCI approaches, which are, as we know, very practical and cheap, and thus allowing for numerous training sessions. This is quite convenient, but on the other side, they suffer for a very limited spatial resolution, which really impairs the specificity of motor regulation. On the other hand, recent works have tried to apply the neurofeedback paradigm using fMRI. fMRI, as we've seen, guarantees a more specific identification of cortical targets. However, it is a very expensive, cumbersome technology, and this strongly limits the number of training sessions. So this this was one point. So the two different approaches, unimodal approaches, used for stroke rehabilitation. It is important, in previous presentation has already been mentioned, that EEG and fMRI also measures different aspects of brain functions. So here in this schematic, I tried to summarize the process of brain activation, and neural activity, so electrical activity, is followed by a series of events, for instance, blood vessel dilatation, changes in magnetic properties which are detectable by magnetic resonance imaging. So we can measure brain function at different latencies and with different techniques. In this curve here, which represent the bold response, the hemodynamic response, the fMRI is sensitive to, we can see that actually the peak in response occurs a few seconds after the actual stimuli. And as Stefan described, these different aspects, the hemodynamic aspect of brain function and the electrophysiological measurements are only partial observation of the brain function, which is way too complex, but we are the underlying hypothesis that combining them could improve our understanding of brain activity. And this is another way to see on a spatial and temporal resolution graph where EEG and the fMRI are located. So EEG is sensitive to the electrical activity of the brain. So it is quite fast activity, and EEG has an excellent time resolution, but as electric signals from the superficial layer of the cortex have to spread through head tissues to reach the scalp, as a limited spatial resolution. The fMRI is on the opposite side of this graph, and it shows specific spatial resolution and limited temporal resolution. So this other technology here, magnetoencephalography or near-infrared spectroscopy, occupies intermediate position on this graph. But EEG and the fMRI really show high complementary and can guarantee, if combined, a high spatial and temporal resolution. So this was really the starting point of the project Hemisphere, hybrid and simultaneous EEG and fMRI neurofeedback for brain rehabilitation. And the general objective of this project was really to make full and enhanced use of neurofeedback by integrating these two modalities, EEG and fMRI. Now this is quite easy to say and very difficult to implement. It's quite challenging to combine EEG and fMRI, and this is even more difficult when this integration has to be performed in real time for neurofeedback. So it took the effort of the previous generation of PhD and postdoc to implement the bimodal neurofeedback platform and test it on healthy volunteers. And it's only since 1 year, more or less, that we started to apply this platform to test the efficacy of neurofeedback for stroke rehabilitation. So here I give a a pictured overview of the environment where the platform for neurofeedback is installed, and is the Neurinfo platform at the CSU of Rennes. That makes use of really cutting-edge technology. Firstly, a 3 Tesla Prisma scanner, then MR-compatible EEG cap provided with 64 channels, and we are also purchasing a near-infrared spectroscopy system in order to integrate the three modalities in the future. We also have a set of hardware solutions that for synchronization and the display of the neurofeedback. And this is this is a schematic view of the platform. So you can see the EEG unit, so the EEG sensor, the 64 channels EEG cap, which is installed inside the fMRI unit. These two recording units are linked to a neurofeedback control system, which is responsible of synchronizing the data acquisition, pre-processing data, and calculation of the neurofeedback score, and finally visualization of the neurofeedback score to the subject by means of a screen, LCD screen inside the MR bore that the patient can see by looking a rear-facing mirror inside the scan. But the neurofeedback control unit is way more complicated than this. This is a more detailed schematic, and is just to give you an idea of how complex a system for real-time neurofeedback integrating EEG and fMRI can be. This is quite a unique system, is unique in Europe, and is second only to the system of Zotev in the US that applied integrated neurofeedback for psychiatric rehabilitation. And these are, yes, more features of the platform, but I also have a video of the installation. Yes, so typically the 64 EEG cap is installed, then impedance of EEG electrode is checked for safety reasons, but also for the sake of quality of recording reasons. The subject is installed in the MR bore, and instructions are given, and neurofeedback is monitored together with EEG signal and fMRI signal during the neurofeedback training procedure. So the platform that I've just described has been tested in the past years on healthy volunteers with two studies in particular, involving in total 30 healthy subjects. The first study was really to test feasibility of such a system, and also showed that bimodal neurofeedback is more engaging, involves more extended brain area with respect to the unimodal neurofeedback, both EEG and fMRI. And a second study also investigated the efficacy of the visual metaphor, so the bidimensional or unidimensional metaphor. And after that, next step was to test this procedure on stroke patients. So I'm showing here results of a very small pilot study on 4 chronic stroke patients with left hemiparesis. The protocol involved first, of course, a motor assessment, then bimodal neurofeedback sessions, followed by 3 unimodal neurofeedback sessions with EEG only, and finally a last bimodal neurofeedback session, followed by a motor assessment. So I've described the platform for bimodal neurofeedback, while for unimodal neurofeedback we used a more practical solution. It was a Mensia modulo EEG solution with 8 EEG channels that was intended to be practical, easy to install for a high number of training sessions. As I said, the aim of this pilot study was firstly to test the feasibility of such a protocol on stroke patients, which is not obvious. Stroke patients have often motor problems, so to install them in the MR bore is not easy. So really was a feasibility study, but we also wanted to test if neurofeedback was in any way efficient for those patients. Yes, in this slide I tried to summarize a bit the methods regarding the choice of the target for the fMRI calibration, and the choice of the EEG filter for the EEG calibration. So here, as an important point for any kind of brain rehabilitation training is the choice of the target. We've discussed it extensively this morning about psychiatric application. In the case of a stroke, we know that the best target for recovery is the primary motor cortex. However, for most severely affected patients, it is the supplementary motor area may represent an important target, may help at least at the first stage of the recovery process, the relearning pattern. And also we found, and also literature review confirmed that supplementary motor area is an easier target to reach, is easier to activate than primary motor area. So in this pilot study, we also test this multi-target dynamic training strategy that in the first session gave more importance to the activity from the supplementary motor area, and finally rewarded more importantly activity from the primary motor area. For the EEG calibration, we used a quite simple approach. I didn't mention that before the actual neurofeedback training session, we have a calibration session where we ask the patient to perform motor imagery without feedback in order to collect some data and estimate those regions of interest for fMRI and an optimal filter for EEG. So for EEG, we consider only 18 electrodes over the motor area and estimate the SCP filter, common spatial pattern filter, from this first 5 minutes long session. Those calibration features are then used during the neurofeedback training session to calculate the actual neurofeedback score that is fed back to the patient, and that is each update equal to the mean between the fMRI and the EEG score, knowing that EEG is updated 4 times per second because it's a faster measure, and fMRI every second. And here I am showing some results. These are results from one of the 4 patients that particularly efficiently upregulated its primary motor area after the neurofeedback training. So in this first line, we have the actual neurofeedback scores that were shown to the patient in the first session in orange and in the last session in blue. Here, the schematic on the left really is what the patient sees during the what the patient is fed back during the neurofeedback training, and we see that the activation of the ipsilateral motor area was much regular and higher during the second session. The interval that you see are standard error across neurofeedback training sessions. And so the patient upregulated in the final session is activity in the primary motor area. We can see it. I'm sorry if it's probably not big enough, but also from the bold activation maps in the ipsilateral side of the brain. And while these last slides show results for bimodal and unimodal neurofeedback sessions, on the right-hand side we have in the bar plots the event-related desynchronization, which was positive for all the unimodal sessions and involved bilateral electrodes, while EEG regulation inside the MR bore was more difficult to achieve. This is a main limitation of combined EEG and fMRI, as Stefan was mentioning. Is very challenging to obtain a very high quality EEG recording in the MR bore, and therefore its regulation is limited. But with this design, we were hoping that during the bimodal neurofeedback session, the patient could develop a strategy based on the richer and information he was receiving from the combined EEG and fMRI system, that then he could transfer at the unimodal sessions. These are results for the 4 patients. So the first column shows motor score, Fagermeyer score. These attest for the motricity of the affected limb. And in the second and third columns, activity in the ipsilateral primary motor and supplementary motor area cortex. So 3 out of 4 patients managed to significantly upregulate their activity in ipsilateral motor area. This is quite, let's say, encouraging, even if from a small sample such this one, because they were chronic patients, at more than 1 year post-stroke. And so for them, no spontaneous recovery is expected, and with a protocol that lasted only 1 week. More importantly, as also the scatter plot on the left bottom part of the slide indicates, this improvement, this upregulation of the ipsilateral motor area was associated with an improvement in the motor score. So for 2 of those patients, the motricity of the upper limb improved after 1 week of neurofeedback training. These are, as I was saying, preliminary results from a very small sample, but they show feasibility of this protocol, and they are encouraging in view of the randomized control trial we are actually collecting data for. This is a more ambitious study that will involve a training protocol lasting 5 weeks, alternating for the control, for the interventional group, bimodal and unimodal neurofeedback sessions, while the control group will only have sessions of motor imagery training without feedback, in order to really test for the specific effect of neurofeedback. This study is quite demanding in terms of resource deployed, of cost, and of time. We will probably discuss some results next year, but for the moment I conclude my presentation by acknowledging the work of all the people that contributed to the Hemisphere project and are still contributing, in particular Mathis Fleury, Dr. Simon Butet, that are actually working every day on the randomized control study. And I thank you all for your attention as well.